Hello everyone! This section is for nutrients uptake. After learning this section, you should be able to compare and contrast passive diffusion, facilitate diffusion, active transport, and group translocation, and provide examples of each. So, what are the common features of nutrient uptake by bacteria? Bacteria can only take in dissolved molecules. Uptake mechanisms are specific. That is, the necessary substances and not others are acquired. It does a cell no good to take in a substance that it cannot use. Bacteria are able to transport nutrients into the cell even when the concentration of nutrient inside the cell is higher than the concentration outside. Thus, they are able to move nutrients up a concentration gradient. That is important because bacteria often live in nutrient-poor habitats. There are four major transport mechanisms for bacteria to uptake the nutrients. The first is passive diffusion. The second is facilitate diffusion. The third are primary and secondary active transport. The last is group translocation. So first, let's talk about passive diffusion. Passive diffusion, often called diffusion or simple diffusion, is the process by which molecules move from a region of higher concentration to one of lower concentration. That is, the molecules move down the concentration gradient. The rate of passive diffusion depends on the size of the concentration gradient between a cell's exterior and its interior. Look at the picture on the right, such as oxygen, carbon dioxide, water can be transferred into the cell through passive diffusion. The second is facilitate diffusion. During facilitate diffusion, substances move across the plasma membrane with assistance of either channels or carriers. The rate of facilitate diffusion increases with the concentration gradient much more rapidly and at a lower concentrations of the diffusion molecules than that of passive diffusion. So what are channels or carriers? Effectively, they are transport proteins. Bacterial cells employ a variety of transport proteins in their uptake mechanisms. These important proteins are embedded in membranes and can be classified into several major types, are channels and carriers. Channels, as their name indicates, they are proteins that form pores in membranes, through which substances can pass. They are often involved in facilitate diffusion. Channels show some specificity for the substances that pass through them. But this is considerably less than that shown by carriers. Carriers are far more substrate specific, and carriers are so named because they carry nutrients across the membrane. Then let's say facilitate diffusion. When the transporter is a carrier, the diffusion rate reaches a plateau above a specific gradient value because the carrier protein is saturated. That is, it is transporting as many solute molecules as possible. Look at the picture. The blue line shows passive diffusion, and the red one shows carrier facilitated diffusion. The plateau in this line represents the saturation effect that is seen whenever a carrier protein is involved in transport. Facilitated diffusion is truly diffusion, even though a transport protein is involved. A concentration gradient spanning the membrane drives the movement of molecules, and no metabolic energy input is required. See this picture? It is a model of facilitated diffusion. You can see the carrier. Carrier protein in its outward-facing conformation. Then it binds solute. After binding solutes, the carrier protein in its inward-facing conformation. Here, it releases solute into cell. After releasing the solute, the carrier changes its formation. Then, the active transport. Active transport is the transport of solute molecules to higher concentration. For example, against a concentration gradient, with the input of metabolic energy. 3 types of active transport are observed in bacteria. The first is primary active transport. The second is secondary active transport, and group translocation. These 3 different transport mechanisms differ in terms of the energy used to drive transport and on whether or not the transported molecule is modified as it enters. Active transport resembles facilitated diffusion in that it involves carrier proteins. Recall that carrier proteins bind particularly solutes with great specificity. Active transport is also characterized by the carrier saturation effect at high solute concentration. Nevertheless, active transport differs from facilitated diffusion in its use of metabolic energy and its ability to concentrate substances. Metabolic inhibitors that block energy production inhibit active transport, but do not immediately affect facilitated diffusion. So let's say primary active transport. The primary active transport is mediated by carriers, called primary active transporters. They use the energy provided by ATP hydrolysis to move substances against a concentration gradient without modifying them. Primary active transporters are uniporters. That is, they move a single molecule across the membrane. You can see the uniporter on the picture. We can say the uniporter transfers the substance only from the outside to inside. For example, ATP binding cassette transporters are important primary active transporters. We also call them ABC transporters. Then the secondary active transport. Secondary active transport covers the potential energy of ion gradients to transport of substances without modifying them. Secondary active transporters are co-transporters. You can see the co-transporters on the picture. They move two substances simultaneously, the ion and the substance. When the ion and other substance both move in the same direction, it is called symport, and the co-transporter, we call it symporter. When they move in opposite directions, it is called antiport, and the co-transporter, we call it antiporter. Bacteria often have more than one transport system for a nutrient. It can be seen with E. coli. This bacteria has at least 5 transport systems for the sugar galactose, 3 systems each for the amino acid glutamate and leucine, and 2 potassium transport complexes. When several transport systems exist for the same substance, the systems differ in such properties as their energy source, their affinity for the solute transported, and the nature of their regulation. This diversity gives the bacteria an added competitive advantage in a variable environment. At last, let's talk about group translocation. The distinguishing characteristic of group translocation is that a molecule is chemically modified as it is brought into the cell. The best known group translocation system is the phosphonol pyruvate. It is sugar phosphortransferase system, PTS for short, which is observed in many bacteria. Look at the picture on the right. The PTS transports a variety of sugars, while phosphorylating them, using phosphonol pyruvate, PEP, as a phosphate donor. PEP is an important intermediate of a biochemical pathway used by many bacteria to extract energy from organic energy sources. PEP is a high-energy molecule that can be used to synthesize ATP, the cell's energy currency. However, when it is used in PTS reactions, the energy present in PEP is used to energize uptake rather than ATP synthesis. The transfer of phosphorate from PEP to the incoming molecule involves several proteins and is an example of a phosphorylase system. In E. coli and Salmonella, the PTS consists of 2 enzymes and a low molecular weight heat-stable protein. We also call it HPR. A phosphate is transferred from PEP to enzyme 2 with the aid of enzyme 1 and HPR. Enzyme 2 then phosphorylates the sugar molecules, as it is carried across the membrane. Many different PTS exist, and they vary in terms of the sugars they transport. The specificity lies with the type of enzyme 2 used in the PTS. However, enzyme 1 and HPR are the same in all PTS used by a bacteria. Okay, that's all for this section. Here is a word list for you.